Good afternoon, everyone, and hi from the Izmir, Turkey. Welcome to talk of the ISCB Regional Student Group Turkey's webinar project by Infonet. As you already know, today's talk will be given by Dr. Ezgi Karaca from Izmir International Biomedicine and the Genome Institute. I'm a master's student here, and she's also my supervisor, and she will be talking about the integrative modeling of the biomolecular complexes. We will have a question and answer session after the talk, so if you have any questions, please click the button with the hand icon on the right-hand side of your control panel and then type your question to the chat box. Now, I would like to invite Dr. Ezgi Karaca to give us her talk.
Good afternoon, everyone. At least for us, it's good afternoon. I hope that you all are hearing us well. If that's not the case, you can also contact us via the chat panel. Today I will talk about integrative modeling of biomolecular complexes. It's also a kind of journey that brought me up to this point, starting from my postdoc and then starting with my PhD, and continuing throughout my postdoc. Maybe this will also give a sort of example to you which kind of path to follow in your personal academic career as well. So to start with, as Tülay mentioned, we are located in Izmir. So we are the first and only international life sciences institute of Turkey. As you see, the mark over there the picture in the middle is our institute. And the mark from the institute is more or less where our offices are located. And you see on the right-hand side, Tülay, one of my master's students, and on the left-hand side, Deniz, the other one. After this brief introduction of the institute and its location, let's start with the scientific part. As we all know, life is operated by molecular machines, so lots of tasks are carried out by tiny machines, and we see a collection of examples over here, let's say ribosome RNA polymerase or a GROAL complex. With different tools, of course, we can try to deduce the function of these and the working mechanism of these complexes. But in order to elucidate a full understanding of the functioning of these complexes, we need to understand and acquire the three-dimensional structures of these complexes. So there are lots of ways to acquire the 3D structure of macromolecules, macromolecular complexes. Of course, the most accurate ones are the experimental. The most accurate ones are the ones coming from experimental methods. With X-ray, NMR, or cryo-EM techniques, we can get numerous structures on the complex formation or on domain-domain interactions or more smaller cases like peptide-mediated interactions. But in case the complex that we are studying eludes these techniques or it just gets too costly or too time-consuming, then in this case, we can also consult computational methods. So among the computational methods, to get the structure of complexes, we can make use of the homology of the cases for which we have structure. Or alternatively, in case we know the individual structures of biomolecules, but we don't know how the interaction takes place, we can use biomolecular docking. Or alternatively, we can gather all the available information on the complex that we are studying, which can come from biochemical experiments or biophysical experiments. And these experimental data might be not enough to calculate the structure, but they might be still good to guide the modeling. And this type of modeling we call as integrative modeling. Sometimes it's also called integrative structure determination. Integrative modeling has two aspects. One is experimental, the other one is computational. In the experimental aspect, as I was mentioning, we are trying to gather as much information as we can from experimental methods, which can be, here you see some examples, for example, from solution NMR, or from cross-linking experiments, or from EPR techniques. And the idea is to gather all this information and to merge them, or to feed them, into a computational method in order to translate this information into accurate solutions of biomolecular complexes. So among all these methods, some of them can give information on the shape of the complex, some of them on the proximity of monomers with respect to each other. But among all these complexes, I will just briefly go over the solution NMR and SAX-SANS techniques to be able to give you a highlight on the type of information that we can get from these methods. Firstly, we'll start with NMR. From NMR, we can get proximity information, and that information can be at different levels. For example, with the chemical shift perturbation, we can get interface information. So this works in this way. We have one monomer in our system, and then in our experiment, we titrate the other monomer, and we see the change of we trace the change in the chemistry of the monomer that we are already studying. And with this, we kind of assume that, okay, whatever is changed should be at the interface. So with this technique, we can get an interface information. Alternatively, from NMR, again, the technique that is in the middle, we can get short-range interaction information. This is pretty specific. It's interatomic, so we know which atoms should be in close proximity with each other, and these distances usually goes up to 5 to 6 angstroms. Alternatively, as the third option, again with NMR, we can use PRE experiments. We can attach a paramagnetic label on one of the monomers that is present in our complex, and we can trace the effect of the paramagnetic label on the other one, on the other monomer in the complex. So this sort of gives us a more like a medium to long range information up to 30 to 35 angstroms. So assuming that we acquired our proximity information from NMR experiments, we can now switch to an example of a shape information. So that shape information can come from scattering experiments, small angle scattering experiments. In the first case, the one you see on the left-hand side, this is the case where we get a scattering profile as a result of spraying X-ray beams onto our sample. So with this profile, we can get an information on the overall shape of the complex, as the red circle indicates. Alternatively, instead of using X-ray source, we can use a neutron source, and in this case, we can use different deuteration levels of the monomers of the complex that we are studying. What does this gives us? This matches some monomers of the complex with the solvent, so that we can get the shape information only on one part of the system. So this experiment highlights the shape of a subcomplex within our complex. And that is also a very useful information, and how useful it can be, you will see in the last bit of my presentation. So this was the experimental aspect, so some highlights on the experimental aspect. As I said, one can use different types of information as well, but I'm not going to go into the details of the other experiment types. Now let's switch to the computational aspect. So the computational aspect is composed on 2 layers of complexities. Let's put it in this way. So the first level is a sampling. Excuse me. Can everybody hear me? Maybe I'll type a message here. Perfect. Okay, super. Thanks for the responses. So, as I said, the first level of sort of the first stage in the computational aspect is sampling. Sampling means to enumerate all the possible solutions that obeys a certain criteria. And in the case of integrative modeling, usually this criteria is minimization of a target function. So you see a target function over here, which is a summation of two terms. The first term is the force field term. The second term is the experimental term. So the force field term denotes the properties of biomolecular interactions. I will get into the details of this in the upcoming slide. And the experimental term tells us how our models agrees with the input data that we fed into the system, into our program. So as you see in this figure, in the case of integrative modeling, we don't use like a sort of global sampling, in a sense that we don't sample all the possible solutions. Instead, we sample the solutions that is in the direction of the force field and the experimental data at the same time. That's why we are going through a sort of restricted funnel. When all the sampling is done, we would left with lots of solutions. And in this case, we would need to pick some of them as the biologically relevant ones. And that stage is called as scoring. So these are the sorts of brief descriptions of sampling and scoring. That is the main, which are the main pillars of the computational aspect. And now we will see the details of them. Okay. So let's start with the sampling and the force field term in sampling. This sort of term is composed of physical formulas that explains interaction phenomena in a very simple way. In the case of biomolecular interactions, we usually don't take into account the energies or the terms that comes from bonds, angle, torsions, etc., because we already assume that we know the geometry of the monomers that makes up our system. But we would still need to calculate the electrostatic and sort of complementarity interactions. And these we calculate by using a mathematical formula. So this is the definition of the force field, where we try to describe the chemistry of the system or the physics of the system with mathematical formula. Then coming to the experimental term in the target function, there are different ways to calculate this experimental term, but the most common way that is used for this subject is to have a sort of harmonic function. So what does harmonic function mean? It means that we would have a term that is called rcalc and another term that's called rdata. So we are seeking the agreement between this rcalc and rdata. So rdata is the initial data that we collected with experiments, and the rcalc is the one that is back calculated based on the model that we are calculating at the moment. So we want them to be as close as possible, and the harmonic nature of this equation comes from the squared part of the difference. And the harmonic potential, this harmonic equation, can be expressed in different terms. For example, you can say, okay, I would like to have atom A and atom B within X distance, let's say within 5 and 10 angstrom. Then in this case, you would have a flat bottom, as you see in the first row in the figure. Or alternatively, you can give an exact measure and say, like, okay, I want this property to be really like 5 angstrom. Then that's the points potential and the evaluation of a point potential you see on the bottom of the figure. So this is the experimental aspect. Just to sum it up, we have force field terms as expressed in physics-based mathematical formula, and we also have a sort of harmonic potential based on the agreement of the data with the models that we are calculating. So with this, we make up our target function. But how are we going to minimize it? As I mentioned, the target function is minimized to be able to sample new solutions. There are different ways to minimize the target function, and you see the 3 most prominent examples over here, which are preferred for different purposes. For example, you can use, let's say, rigid body minimization, where your monomers are treated as rigid bodies, so you would only have translation and rotation of your monomers with respect to each other. This is not going to be very accurate, but on the other hand, it's going to be fast because you won't have too many degrees of freedom in your system to deal with. Or alternatively, let's say your rigid body minimization brings you from a black point to the green point, but alternatively, you can also use simulated annealing to bypass higher barriers, in which case you heat up your system to an inaccessible temperature so that the system can adjust itself more quickly than a standard room temperature. Then you start, after having some simulation time, minimization time, then you cool down your system. Or alternatively, if you think that you already bypassed your energy barriers and you want to sample what is out there in the world that you already are, then you can also use molecular dynamics to be able to see how your complex, how the monomers can move with respect to each other within a certain energy level. So this makes up the minimization part. So with this, the overview on sampling is finished. Now let's come to the scoring. Okay, we sampled, we generated, let's say, 5,000 structures, 5,000 complexes. What are we going to do with them? How are we going to decide which one is more accurate than the other one, right? That's also a big problem to solve. So the standard scoring functions that are out there, they are usually using some common terms. So either they use force field terms, let's say electrostatic interactions or van der Waals interactions, or they use the buried surface area because it's assumed that if the buried surface area is larger, then the binding is more stable or more prominent. Or alternatively, one can go to databases and then analyze available interactions, and from there deduce some interaction propensities. That is also used as a scoring term. But none of these are able to address very well the state of binding of a complex. Why? You see the reason why in the figure on the slide. And whatever we calculate, it becomes the function if it becomes a property of the bound state. So we know the bound state. We can calculate the properties of the bound state, but we don't know how the unbound state behaves. That's why with these terms, we can comment on the binding properties of a bound complex maybe, but we cannot comment on the binding affinity of the complexes very accurately. As for this, we would also need to calculate the energies of the free states. So if this is the case, what should we do then to be able to really be proper on scoring? We can, for example, use a sort of extra layer of filtering based on an available experimental data that can be a mutation information, for example, which says that, okay, if you mutate residue X and residue Y, we don't see any binding. And if you find these residues to be at the interface in your models, then you can say that, okay, this should be my solution. Or alternatively, we can use the experimental energy term, which measures the agreements of our model with respect to the experimental data. We can incorporate this term into the scoring function to make it more accurate. In a sense that if you would like to have a more accurate scoring function, we should try to get some information also from experiments or from literature that's already published on the system to be able to see that we are doing well. And okay, we sampled, we scored things, but how are we going to evaluate our models? So there are two measures on the evaluation. These are accuracy and precision. And this DARTS slide kind of summarizes these two concepts pretty well. If you hit the dart at the center, then you are both accurate and precise. Alternatively, you might have selected solutions that really look like each other, so they are precise, but you might not be at the center of the dartboard, so this means that you are not accurate. Or you might have all scattered solutions. This means that you are neither accurate nor precise. So how are we gonna understand whether we are accurate or precise? So the precision is measured with clustering. So you can use a similarity measure to group your solutions. That will tell you how precise your complexes are, how precise your models are. The standard measure in clustering is usually to use root mean square deviation as a similarity measure, but it can be also something else as well, which another measure that you know that is important for your system. And coming to accuracy, unfortunately, without having experimental validation, it's almost impossible to comment on the accuracy of the model. So which also highlights that actually modeling is not a sort of standalone practice. Modeling is a part of a circle which should be always fed with experiments, and with models we should always propose new hypotheses for new experiments. Okay, so for the accuracy testing, there is a community-wide experiment that is being organized already for quite some time. It's called CAPRI. It's analogous to CASP, but instead of predicting protein structures, in this case, we try to predict the interactions between monomers. And if you are curious which groups are participating, which programs are participating in CAPRI, you can go to its page on EMBL-EBI. And you can see the community papers to be able to decide which program performs in which manner. Okay, so this was sort of an okay overview on the background of integrative modeling. So, of course, when explained like this, it feels like all the problems are solved. We know which minimization algorithm to take, or we know how to formulate our scoring function, etc. But of course, as in all of the modeling exercises, things are not that bright in the case of integrative modeling too. So in this case, we have 4 major challenges that are noteworthy to talk about. The first challenge is to be able to deal with sparse data. What does sparsity mean? It means that you have information only on one part of the system. So the information is not homogeneously distributed, and that's a minus. The other challenge is that we need to deal with multiple type of multiple molecules that are heterogeneous in nature. This means that we should address different levels of degrees of freedom for different types of molecules, like protein, RNA, or small molecule at the same time, because the big molecules, they contain all these sorts of molecules. And then the third one is to be able to address conformational changes that are happening upon binding. And finally, as I mentioned, is the scoring problem, which is finding the biologically relevant solution among all these structures, among all the models generated. Considering the fact that the integrative modeling tool that we should use is general, then we have 3 prominent methods. So there are other types of integrative modeling tools as well, but these are usually fine-tuned for certain types of experimental data, or for certain types of modeling problems. But all these 3 that you see on the slide, they are generic, and they can address lots of different scenarios in a quite nice and elegant way. So the first one is IMP, the other one is Rosetta, and the third one is HADDOCK. I'm gonna focus on HADDOCK from this point on, which was also the major focus during my PhD. Maybe some of you already know about it. HADDOCK is abbreviated for High Ambiguity Driven Docking. And this means that we can incorporate experimental or predicted, let's say, bioinformatics data into our docking protocol, into the complex formation. And this data can be in the form of interface distances, orientation, etc., as I was mentioning in the first slides. And the good thing of HADDOCK is that it is used independently of the developers. So it's very commonly used by structural biologists. And you also see a proof of this that if you go to Protein Data Bank, you will see that there are more than 100 complexes that are deposited into PDB, and these complexes are calculated with HADDOCK. So the reason for this is that it has a web server, and the web server is quite user-friendly, and that also makes the program accessible to broader communities. Okay, so the docking protocol of HADDOCK very briefly starts with separation of the monomers and random collision of these monomers with respect to each other along the direction of the data and the force field. And then the good scoring solutions are submitted to the next step, where the interface of these good scoring solutions are refined to be able to address small to medium scale large conformational changes that are happening upon binding. And finally, all these solutions are refined in water, and then they are clustered. This was sort of the sampling phase, and in the case of very different minimization functions are used. And in the case of scoring, at the end of each scoring stage, a collection of terms are used, which also follows the scoring slide that I showed you. So here we have force field terms or buried surface area, etc. So the exact weights or the details are not that important at this point. So just to remind you the challenges again, so we need to deal with sparse data, we need to deal with multiple heterogeneous molecule, large conformational changes upon binding, and we also need to pick the biologically relevant structure among the pool of complexes that we produced. So I will, from this point on, I will highlight the solutions that we developed throughout the years to deal with these mentioned challenges. The first example comes from a scoring exercise. So in the scoring exercise, we demonstrated that if we have a SAX data already at hand, and if we incorporate the SAX data to our standard scoring function, we can improve the accuracy of our scoring. So that is sort of the first example that I can show. The other example is the other example that we developed regarding addressing these challenges is the fact that the current web server distribution of HADDOCK can deal with simultaneous docking of up to 6 monomers. And the monomers can be in any combination, so they can be the same monomers or different monomers. And you can also impose a symmetry between these monomers. So that's also a plus. And a while ago, we demonstrated that by using this sort of multi-body protocol and by using literature data on some complexes which come on conservation and mutagenesis information, we can back calculate very accurately a number of heteromeric and homomeric complexes. So here you see, here what you see is the gray structure is the crystal structure, and the pink one is our proposed model. And you, in angstroms, you see the reported accuracy. So as you can see, they agree pretty well. Coming to another method which we developed to deal with large conformational changes upon binding, that relies on a sort of divide and conquer strategy. We said, okay, if we would like to address large domain motions upon binding, maybe we can treat the rigid domains that move upon binding as separate bodies. And for this, we proposed this flexible multi-domain docking, FMD. So the idea for this case starts with definition of the hinge regions by using a hinge predictor, hinge prot, so that this part is totally blind. So this hinge regions define where we are gonna dissect the rigid monomers, so the sort of cutting point, and then we cut the we cut a flexible monomer at this hinge region. Afterwards, we define an imaginary distance restraint for the imaginary peptide linkage, or let's say for the removed bond. Then by using the multi-body docking option of HADDOCK and by using interface information, we demonstrated that we can address conformational changes that are as large as 20 angstrom. And that's already quite a nice achievement. Okay, so these were some examples on the methods that we developed and that was proposed to deal with the challenges of the integrative modeling field. Now I will switch to a real case application where we use real experimental data, where we collaborated with experimentalists actually, to show you how we can how we perform integrative modeling on a real case scenario. So this example is on the ubiquitin cleavage preference of a ubiquitin cleaving enzyme, Josephin. So for this case, it was known that Josephin cleaves multi-ubiquitin chains, but the preference of Josephin was unclear. So is Josephin cleaving ubiquitin chains that are linked through lysine 48 or lysine 63? And in this project, as starting information, we had binding site of the diubiquitin, so 2 ubiquitin monomers on Josephin. This came from mutation and also NMR chemical shift perturbation experiments. We also know, of course, the catalytic side of the enzyme, as the enzyme is functionally characterized. So by using only these two information, we demonstrated that if we have the 48 type of linkage, then the frontal ubiquitin can direct its C-terminus to the catalytic core of the Josephin enzyme. But if that's not the case, if the linkage is made through the 63 linkage, then topologically the frontal ubiquitin cannot reach the catalytic triad. And this was also validated with wet lab experiments. So this, with all this work, sort of summarizes my PhD work. And when I was moving out for my postdoc, I was thinking like, okay, I have developed lots of nice tools, but these are rather valid for small scale systems. But if we would like to go for really large molecular machines, let's say ATP synthase or proteasome, we should have a different type of approach. So this idea was the central idea of my postdoctoral work, and the protocol that we deduced to determine the structure of molecular machines actually starts with a separation of our initial experimental set into two orthogonal sets. Usually what is advised in the literature, and also what we are advising as well, is to collect complementary data, let's say proximity and shape, and use proximity for sampling and use shape for scoring. So the validity of this has been proved multiple times. So this is also what we are offering here, what we are proposing here. So the idea is to get proximity information first and then calculate all the possible solutions that are accessible along the direction of the proximity experiments, proximity data that are coming from experiments. And then after having this sampling, which is called as global search, we score our solutions. But instead of using a physics-based function or a sort of knowledge-based function, we consult here statistics. So what we are doing is we analyze all the experimental energy term of the solution, so the agreement of each solution with respect to the experimental data that we inputted, so the proximity data. And we try to see whether in the whole population that we generated, there are a bunch of solutions that behave differently from the rest of the population. What does this mean? The rest of the population, as you see the box and whisker part of our plot, that is kind of the Gaussian part. So these are rather random samplings. And the ones that are pop up here as outliers, these are the differently behaving ones. And here, if we cannot generate a differently behaving structures, then this means that either our data is not enough or our sampling is not enough. So instead of using a standard function, incorporating a statistics-based measure here also puts a sort of first layer of validation on our protocol. And that is sort of unique compared to the other protocols that are available in the literature. So if we have some outliers, then we isolate these outliers and we sample around these outliers further. Here we use exhaustive simulated annealing, different than the standard protocol of HADDOCK. So we continue sampling over here. And when this is done, we take our shape data and then we score everything with respect to our shape data. If we find a correlation between the conformational similarity, so between how the solutions that we generated are similar to each other and their fitness to the initial shape data, so if we find a correlation over here, as you see in the slide, then we isolate the first cluster as the biologically relevant, probably biologically relevant cluster. So this is sort of the highlight of the overview of the method. And as a proof of principle example, I will go over this very briefly, which is a case for which we already know the solution. This is a coarse splicesome complex, which we know that it's made up a ring structure through which an RNA is passing, which is going to be spliced. So we know this information. We also know the interaction size of protein and RNA. In this case, we assume that, okay, we will have PRE data, so there's medium to long range distances from NMR, and combination of this data and feeding them to our program, we can see on the left-hand side the outliers that are generated and how the outliers are looking on the right-hand side of the slide. So we see a structural similarity on the x-axis and this experimental energy on the y-axis. As you can see, there are structures that have similar restraint violation energy, but very different RMSDs. And if we continue, if we're taking this example to local search and continue sampling, we see that we can get way smaller energies. So that's very good, but very different solutions from each other. And these different solutions can be separated from each other by using SAX data. So we see that a bunch of the solutions, they don't fit the SAX data, but the other good portion fits very nicely the SAX data, and it also reproduces the starting structure. Okay, so I will skip this slides. And so this was a simulated case, and we have a validation example that is coming from experiments. There's coming from real experimental data. So in this case, we had we took data from another published method, which used cross-linking, mass spec cross-linking distances. These are medium-range proximity distances and the EM shape of the whole RNA polymerase. And in the end, with this data, we showed that our solution, M3 solution, that's how we call our method, resembles the crystal structure pretty well compared to a previously published method. So this sort of gave us a confidence that we can perform well with different types of data as well, not only with NMR SAGs, but also with cross-linking and cryo-EM data. And the final example is on a complex that is responsible on ribosomal RNA methylation. So a methyl group is attached on the ribose moiety of the RNA. So this is a very large and complicated complex. So I'm not going to go into the details of the complex. I will very briefly mention about it. The core of the complex is made by a guide RNA. And this guide RNA has single-stranded parts which double strands with the ribosomal RNA to be able to fix the ribosomal RNA for the methyltransferase reaction to occur. This complex is pretty big, it's 400 kilodalton, and the methylation takes place through an assembly making protein, that is the gray one over here, NOP5. NOP5 takes the RNA recognition RNA recognition protein, the green one, and also the methyltransferase, the blue one, puts them on the guide RNA and sort of leads to the reaction. It leads to the reaction. Sorry. For this case, the complex was unknown. So the assembly of the complex. So we assume that the assembly of the complex was unknown, but the substructures of the system were known. So what did we do? We took these substructures and we recycled a method that was previously developed. The FMD method, which I also mentioned in the previous slides. So we assembled by using imaginary linkages between these monomers, we assembled these subcomplexes with NMR data and with contrast matching sense data. So this is the data type that I was mentioning you, so we can get a shape of a subpart of the complex with this data. So we have RNA shape, the shape of the RNA within the complex in this case. And we also had some other scattering profiles to score our modeling. And in the end, on the left-hand side, you see the structure that was calculated more or less through standard NMR structure calculation approaches. And on the right-hand side, you see the model that we proposed that is calculated with our method. And you see that both of the solutions agree pretty well. So this means that actually by combination of different protocols that were developed up to date for this particular work, that were developed up to date, one could gather them, recycle them, and put them in a different perspective to be able to deal with way more challenging situations. And with this, I would like to finish up my presentation. So all in all the tools that I shown you, or the sort of background on integrative modeling that I shown you, led me to a point where a method called M3 is developed. With this method, I mean, with a similar mentality, one can develop an alternative method as well, but in particular with this method, I believe that we address the major challenges of the integrative modeling fields. And if you are curious of this method and of the other methods, you can go to our group's website, or you can also get the code of the method from my GitHub account. And with that, I hope that I would like to thank you first of all, and for the kind invitation for me to give this webinar. And I hope that you could follow most of the presentation. I know that I had too much information, and it's also not so very straightforward to present without seeing you looking at me, but still I enjoyed it, and I would gladly appreciate to answer your questions.
Thank you, Dr. Karaca, for your talk. Now we can start our question and the answer session. If you have any questions, please write to the chat box so we can check it.
Maybe in the meantime, if you have a question or a point that was not well understood.
I think we can maybe explain. Can we understand the binding affinity from the using the HADDOCK?
So there are. I mean, my expertise is not on. My past expertise, at least, is not on binding affinity, but as far as I follow and as far as I know. The binding affinity prediction is possible for rather rigid binders, so that's rather a sort of solved case. But for the binders which act flexible upon binding, these are rather more difficult to tackle with. So the common approach is to classify cases like, okay, I'm going to work on antibody antigens. Let's see how antibody antigens are binding, and let's see how we can develop a scoring function for this. So that's an approach. Or alternatively, as I said, you classify complexes as rigid binders or flexible binders, and that's also a solution. But unfortunately, there is within the perspective of HADDOCK or within the perspective of other predictors, there is no standard sort of function that we can safely say, like, okay, this can be used to predict the binding affinity of complex X or complex Y. So we always should know what we are dealing with and the capabilities and limitations of the method that we are using. Okay, so Ersin is asking, we know sequence and we want to predict 3D structure and propose potential aptamers for experimentalists. How to tackle this? So in case you know the sequence, but you don't know the structure, then the most common way to address this problem is to use homology modeling. That is something that I mentioned in one slide only, so I didn't go into the details of this method. So one can try to model the monomer by homology modeling and hope that there are homologous structures, there are homologous sequences of your case that have a structure. And after having this, after modeling the structure, the solid way to approach this would be to define the binding region. So where are you going to target? So what is going to be the target region for you on your model? And after that, after having this target region, you could go and see whether you have any similar interactions that you expect from structural databases. And if you don't have anything to predict these things de novo, like from scratch, is not easy at all. I guess you should really know how your system behaves. And if you are lucky, maybe you would have an example already and you would just try to modify this. So I know that I couldn't give a formula, but this is like one of the toughest sort of questions of the modeling field. You're welcome.
If there is no more questions, so I would like to thank Dr. Karaca again for her clear presentation and contribution to our project and the participants for their support. Our next events will be announced on BigMarker and our Facebook and the Twitter page. Please register to Bioinfinite community on BigMarker to receive our invitations when new events are organized. Have a nice week, everyone.
Have a nice week as well. And if you have any questions, you can reach us via the email address that you have on the slide. And please follow these guys. I think what they're doing is great. So see you next time.